One of the central-most questions in developmental biology is how does the embryo, which starts as a single cell with limitless potential, develop into all the complex cell types, tissues, and organs that make up the adult organism? The mouse has been an ideal model organism to address this question, as it shares all the same tissues and organs as humans, but is smaller, develops much faster, and allows for genetic manipulation. One day, it may be possible to recreate entire organs outside of the body for stem cell therapies, tissue replacements, or transplants. But first we need to understand how these organs develop naturally. Which led us to ask, how does the early mouse embryo first start to form tissues and organs? Can we visualize and quantify this process live in an intact embryo, at the single cell level, and watch as cells move, divide, and interact to build the developing embryo? These questions have remained unresolved until now because the mouse embryo presents numerous fundamental challenges to both culture and live imaging. First of all, mouse embryos grow inside the uterus and must be removed from the mother in order for their development to be visualized. Secondly, mouse embryos are extremely photosensitive, and their culture demands precise media, gas, and temperature conditions. Finally, mouse embryos grow rapidly in size and have complex optical properties. They require the perfect environment in order to develop normally. Because of these challenges, it has so far not been possible to image the early stages of organogenesis in the mouse embryo in toto or with single-cell resolution. To overcome these obstacles, we built a light sheet microscope specifically tailored to the needs of and optical challenges presented by the mouse embryo. This microscope includes a custom environmental chamber to maintain optimal culture conditions and can sustain normal embryonic development for days at a time while the embryo is being continuously imaged. Moreover, because the embryo is constantly growing and changing in size and shape, the microscope is designed to automatically measure and adapt itself to the dynamic growth and optical properties of the embryo. This self-adaptation keeps the embryo focused and centered in the field of view. With these new capabilities, we can image mouse development from the symmetry-breaking event of gastrulation through the organogenesis some 2 days later, at the high spatiotemporal resolution required to track individual cell behavior. We can visualize different developmental processes as they occur, such as heart development, neural tube closure, or the formation of the notochord. In order to analyze and quantify these developmental processes and make comparisons between embryos, we developed a computation framework to handle the large and complex image data generated by the light sheet microscope, including a machine learning framework that uses a convolutional neural network to automatically detect cell divisions in the 4D image data. We are able to track individual cells from the beginning to the end of our movies by combining our cell tracking framework, TGMM, with a statistical vector flowing technique that is accurate to within 2 to 3 cell diameters over the entire time series. Using this method, we have generated the first dynamic cell fate maps during mouse gastrulation and can measure cell population dynamics and systematically trace the origins of individual cells or tissue types with an unprecedented level of detail and precision. A quantitative, statistical analysis of normal development requires the comparison of multiple embryos, which in the mouse can be challenging due to the dramatic differences in size, shape, and cell number even between embryos of the same developmental stage. We developed a framework we call TARDIS for registering multiple developing embryos in space and time, which allows us to make direct quantitative comparisons between individuals. By combining our high-resolution data of development for multiple registered embryos, we created an average embryo. This approach reveals the average developmental trajectory of the mouse embryo, quantifies the degree of variability in normal development and the ability of the embryo to adjust to different conditions, enables statistical cell fate prediction, and detailed automated comparisons between normal and mutant embryos. Finally, to facilitate access to this information for the scientific community, we have made all these methods and resources, including the computational framework, the microscope itself, the image data, our reconstructions and analysis, and the average embryo freely and publicly available.